So, to close this first session, we thought it could be useful to continue in the path set by Catherine and Helga to say how science and ethics are interconnected, intertwined in the process of governing the future. Process that we have to forge and be aware of. I would like to start from the idea that these dichotomies, which have been with us for many centuries, have come to an end, really. They are not synonymous, facts and values, nature and culture, science and societies, but they have overproduced. Fox Kelly, an historian speaking about nature or nature, used to say the mirage, the idea of a space between nature and nurture. These spaces have given a structure to our world. For example, at the time of the Royal Society, as Helga said, the representatives of the world, the scientists, and then we had the representatives of politics, and the two things were kept separate. Then we started to see how these two things are interconnected. And now we're going to see some concrete examples about what in one thing contains the other one, all the more so from the very start. And we're going to do that after we had the privilege of addressing you over the last 2 years. We close this trilogy, and I'll try to give a structure to the whole discourse we started together 3 years ago, and this is one of the virtues of this conference, gathering us here all together in this place every year. So we will start from the digital gaze, which has already been mentioned, which captures two essential aspects of science today. First of all, a growing number of biological subjects and functions is treated in the lab as a digital phenomenon. And in these subjects and these functions, we intervene through these digital codes, and we're going to see it from genome to organs to cells. In other words, all the classical levels of organizations of what is alive, as we heard from Giuseppe Pellicci, all these aspects have been digitalized, can be reduced to fractals as a digital datum. Here you can see some of the main aspects to say, from the genome of each one of our cells up to whole ecosystems, all these levels, which are levels of biological organization, but which are also epistemic levels, levels through which we choose to tackle a problem from one level or another level. All these levels have already been digitalized or can be digitalized. Understanding that means understanding the notion of a scale from two points of view, both in Italian and in English. Scale as a level, or the ladder, the level at which we study a phenomenon, the scale at which a certain phenomenon becomes manifest, but also scale in terms of dimension, the dimension of this study. But it also means understanding the depth, i.e. that life can go through different levels which are compatible one with the other because they can be digitalized. But for each level, dimension is important, because what makes the difference is the very scale of the study. How big, how small is the knowledge acquired, the study made. To give a concrete example, this is a study published some weeks ago in one of the most important magazines. The High Definition Medicine is a study which has been published. High Definition Medicine focuses on the idea of precision, wholeness, totality, the individual, customized or handmade or made to measure for that individual, with the idea of a set of functions some of which I've already mentioned, which are open to this digital gaze, which should work in such a way that for each one we have the basic level of functioning on which prevention discourse can be built to arrive at treatment. An idea which is the function of another one, which is becoming very fashionable, which is the evidence of the real world. This idea, instead of understanding the truth in an abstract concept, in a clinical study, for example, you go to real individual through these tools, and this makes the real life an object of a digital study, which can be archived. This goes back to what we said 2 years ago with precision medicine, as Chiara Tonelli said yesterday, to understand exactly where the value gets into play. And we can see it from this initiative launched by President Obama, the Precision Medicine Initiative, which was also given the name all of us, all of us, all of the United States, all of us. It's a pun, it's a joke. Anyway, with some scholars, we have highlighted these three aspects, which are the salient aspects of this effort. How different are the individuals represented in this object, in this initiative, which aims at studying 1 million individuals representing the all of us through all the different levels, scales I mentioned earlier on. Now, very briefly, we are starting from the basic aspect, the genome of 3.2 billions of nucleotides composing our DNA, and about 3 million genetic variables which are unique to each and every one of us. In a collective study, in a collective effort, one of the precursors of all of us, try to focus on cancer, to develop the Cancer Genome Atlas, TCGA, which is going to be a very important resource for all of us. But last year, racial and ethnic disparities were discovered because the sample of this atlas included too many white individuals with very few individuals representing other minorities in the USA. And that had a very clear impact because according to the kind of tumor of cancer you wanted to study, since we have different incidents of tumors in different ethnic groups, the choice of saying what is the level I want to reach in this scale, how many mutations of these 3.2 mutations I want to gather to understand where these cancers come from. For all the cancers which were studied, there was the possibility of detecting mutations which were not very frequent for certain somatic types. This applies, for example, to breast cancer. For example, this was true in some cases, but not true for other cases, for ethnical groups. So the ethnical disparities were transported in the epistemic sphere of this project, unwillingly. So this representation of the atlas, a geographical map with the number of inhabitants. But this is the same world, but in terms of wealth. Just to remind us that the science is not producing neutral atlas, but science, as it happened for cancer, this produces atlas based on certain choices and on certain values and choices. This takes me back to this recent case. You may have heard about this case, which led the Italian Institute of Anthropology to propose to cancel the word the word race from our constitution, because all citizens are equal in front of the law. The idea is that race does not exist in scientific terms. At a molecular level, we know that there is more diversity in the center of Africa than in the rest of the world, and then that you decompose in molecular terms, a race does no longer exist. But race does exist in cultural terms, and this is what we see with the spikes of racism that we have seen in our country as well. In the previous study, those minorities, using the concept of race, may put forward a claim and say we are underrepresented before taking out from the Constitution something which does not exist in molecular terms but which is very present in our society. We must be very careful, because these are very important notions which can be beneficial, which can lead to emancipation. Now, let's go back to what has been said before, last year, for example. Another important frontier which has been opened up and lifted by digitalization, that is the possibility of reprogramming cells and patients, not just physically, but also culturally. This is based on the idea that the development, as represented by Conrad or evolution, was a single direction development, saying that the parts of our body which are used to think or to stand up were decided at the very beginning of our evolution. Now we know that we can go backwards and we can reprogram ourselves ad libitum as much as we want. Here you can see our colleagues in Australia, which can grow a small part of the kidney. We can do it in the lab for some parts of the brain cortex, with the idea that the cell itself is an organ standing by itself. The idea of printing organs, when you print reprogrammed organs or cells, and the cell can be treated as if it were an application, because we are starting to learn about its code. Japan, some years ago, in Japan, a new application was launched, and we see that the ethical values and the economic values are interwoven with the scientific values. And this can be seen here when the first woman, the first human being, is transplanted with the retina-pigmented epithelium and reprogrammed starting from the skin of this woman. There is one woman in the world which has in itself one part of the tissue which has been reprogrammed and harvested by her own skin. And the same thing will be applied to Parkinson, reprogramming the skin to become neurons to be implanted in the brain. So in Japan, this was performed on this woman. And they were about to do the same on a second woman when the study was blocked, was suspended by Riken. We can learn a lot from this story. First of all, in the second woman, which was ready to be operated on, it was discovered these cells reprogrammed from her skin contained 3 mutations, 3 changes in the 3.2 billion letters composing our genome, 3 mutations, the meaning of which was unknown. So the idea was, what can we do now when the scale of our glands goes so much in depth that we can read those 3 mutations without knowing what they mean? Who can convince the woman to accept the risk? Is the government supposed to make the decision? Is the scientific community? supposed to do so. So the scientific study was suspended, but something else also happened in Japan. Japan said, yes, we were pioneers in this highly personalized approach, according to which from the skin I can develop the tissue I want and re-implant it where I want. But Japan as a country believed that it was much better to see what are the Japanese whose immune system was compatible or is compatible with most of the other citizens without being so much personalized, but rather I create a cell data bank covering all the Japanese people living in Japan. So a new law was passed, containing not just a scientific breakthrough, but also a mercantile or ethical, which gave rise to a reaction in nature, for example. So Japan said that you only have to prove that these new cells are safe, and then you can put them on the market, and then you have 7 years to show what happens in real people and to show that if these cells are really effective on real people. This is a really dramatic change in the way in which we arrive at conquering scientific certainties. This is what Cooper and Wobley, which published Clinical Labor, mentioned as the market as an extension of the clinical trial and the consumer as an extension of the patient. Because out of these potential Japanese which are taking these cells which are safe but which have proven to be safe but not yet proven to be effective, because it will take 7 years to say whether they are effective or no. They are patients, yes, but they are also consumers, and strongly so. It is interesting, for example, that at the same time a new article was published in one of the most important magazines about biologists, which carried the title Therapies of the State, which is meaningful, even though the idea of state has broken down in many cases. But nevertheless, what happens if a state gives a therapy, and what makes a therapy a state therapy? And now let's remember what happened in our state when a state of knowledge, or state also in terms of political places certifying knowledge, state of knowledge clashes against a real fake treatment, a fraud, really, when two patients, really sick people, take their own blood and try to spread around their blood to ask for that treatment, a fake treatment, to become a state treatment. That was a fraud, but for many months, the state or Italy didn't give a reply to that. And in some cases, many believed that that was going to become a state therapy. That was a charlatan, really. But in Japan, what happens in Japan? First, you have to prove that it is safe, and then you have 7 years to prove whether it is effective or not. Is that case very much different from this example in Italy? And in this space, in this range, we have to set and to ask our questions. So this goes back to what we said last year. So the puzzle we are putting together. So the idea of civic epistemology is part of the puzzle we have been created in this conference. We spoke about these aspects before. As knowledgeable citizens, we believe that there is a gap between the pathway of knowledge and the way in which the civitas, the citizenry, our fellow citizens, get to understand when something is really confirmed as knowledge. So we are going to have a discussion later on, but the institutional practices by which the members of a society test and deploy knowledge claims used as a basis for making collective choices. How can we get there? How can we get to know things together? Where is trust? Where is the institutional pathway which enable a collectivity, a community, which doesn't know many things, but gets to know something as a community, as a group? What are the ways we have to create this common lodge? Of course, it seems that we are not very good at this, but what can we do to understand that? Now, we can discuss the genome as a draft in the last 10 minutes of my speech. And I would like to go back to the idea we presented in this book together. At the time, we were very much struck by Philips saying that all its products had to be based on the idea of science and simplicity. Here we have a CT scan, and for example, Philips used to say with a normal CT scan, you don't understand what this is. You need a gynecologist to tell you that that is the head of the baby. But now I can give you a 3D image through my new tool, and then you can see by yourself what that is without an intermediate go-between. For example, we said that there could be a case of Venter meeting the founder of Google. Venter saying that I want people getting on the computer and become a scientist with no knowledgeable go-between to explain. And we know in Italy that really this direct acquisition of knowledge is one of the serious problems of the political arena today. So here we have the idea of seeing things to solve the problems. This goes back to the Enlightenment period. We have Diderot from the dream from Rameau's nephew. He spoke about ideology in the 18th century. For example, he discussed when can we understand when an embryo is an embryo. That the answer is take an egg. Once you have seen an egg, then this is what disclaims what has been said by all the theological stories and schools and temples. So we know that ideologies and temples have not been dismantled, and now we know that the discourse and the dispute on these values is still very lively. Also because we have the digital phenomenon in front of us. And I would like to go back to something we said in the past, unlike what Diderot said some years ago in America, the issue of the embryo used for research aims was very lively. The idea is to clone before, for example, doing what I said earlier on, where we can go through the many cellular identities in a very simple. In the past, the same could be done by taking an egg, extracting its DNA, and taking the DNA taken from my skin. For example, this gave rise to cloning, the cloning of Dolly. The problem in doing that is that I would have this cloned embryo from which I wanted to harvest the cell to develop a neuron to be implanted, but I could also implant it in a uterus and clone an individual, a real person. The idea in the States was there was no problem. Let's remove a gene from it, an essential gene from development. So when I develop this entity, it's not a clone, but it's rather a liveless growth, which is useful to arrive at this result, but which cannot give cloned individual. This thing was developed in the bioethical committee set up by Bush, was translated by MIT in a real biological phenomenon where you realize that the value was written in the DNA letters because that gene had been removed. And this was published on an important Nature. And Michael Gazzaniga, who was a member of that committee but against this approach, said, normally we generate a word to describe a biological phenomenon, and here we seem to be tinkering with the biological phenomenon to have it fit the meaning of a word. You may remember this scheme, where on the one hand we say to prevent cloning we need a law, we need a universal sanction, but if we modify this gene, this thing cannot happen technologically speaking, and so the problem is solved. We can do it with genetic engineering without waiting for a law to be passed. But this leads us to CRISPR. It was only 10 years ago, but as Pellicci said yesterday, the last 4 years have been a storm in the scientific world because of this simpler way of engineering an embryo. This is a human embryo which has been engineered and published on Nature 2 days ago in one of the first 3 articles showing the great easiness with which we can modify the genome of a human embryo. 2 years ago, we spoke about the CRISPR-Cas as a phenomenon which was to be expected. While waiting for CRISPR-Cas to happen, the real crux of the matter was the idea that the genome can be manipulated, and not just in the case of an adult, to use it then for genome treatment, but the same can be done on an embryo, which will grow and which will pass on this modification to its children and to its offspring. So the question we have to ask is, why is it that we are using this on human embryos? Why is it? What are the values? What values are used in this kind of approach? Let's read this excerpt, which could be a great promotion of CRISPR-Cas and the use of genetic editing on embryos for the germline as well. It says, we want a country that extends its promise of opportunity to everybody who's willing to work for it. We want to have a nation in which the accidents and circumstances of our birth aren't determining our fate, and therefore born with a particular disease or a particular genetic makeup that makes us more vulnerable to something, that's not our destiny, that's not our fate, that we can remake it. This is the backbone. And Barack Obama said so when he launched the President on Precision Medicine. But it's the same spirit, it's the same trajectory which leads us ahead, which leads us towards that dock. That's another aspect, the economic value. This is a tweet with another study which showed that maybe this mechanism is less precise than what it is thought. And there are many other problems, but this study showed that apart from hitting where you wanted to change the DNA, it could also hit on many other places, thus introducing imperfection. That very day, the actions, the shares of the 3 companies investing on CRISPR-Cas collapsed. There was really a collapse in the stock exchange of these 3 companies, because here you have mutations are specifically induced by this new genetic. Here I can count everything. I can count the mistakes made, the errors made in the DNA, and I count the mistakes, the errors, the drops in the stock market. And here you can see an article which 1 month ago surprisingly proved that it is even easier than what we thought. It is very simple to modify the DNA of the human embryos. But why do we want to do that? That's the issue. And the two international summits that try to guide ethics on this topic in just less than 1 year have already modified their agenda, giving a go-ahead to the modification of the germline, but only for very serious diseases. But in very serious diseases, the solution is that is the pre-implant genetic diagnosis. Couples having a serious disease can select the embryos not to implant those bearing the disease or the gene bearing the disease. Why, if we know that, why are we doing all this? Why do we have CRISPR-Cas? Because CRISPR-Cas can offer us the possibility of introducing in an embryo a genetic variable which is not present in the parents, in the couple, so gives what could not be given otherwise. And here, an unproductive discussion is introduced. Either we are going to have children with blue eyes or intelligent. Of course, the first thing belongs to features which are inherited, and intelligence is much more complex than that. But there are other things. For example, the genetic variable that some of us have, which makes them resistant to pneumococcus infections, or the ApoA, which is another genetic variable which protects from Alzheimer. Out of 100 people in this room, 70% of these 100 people have a configuration which protects them, partially so. Others are completely exposed, and 7% are significantly protected from Alzheimer. These are tangible aspects. Why do we have to introduce the engineering of an embryo, or not to do so in an embryo, to offer these two potentialities? This is not allowed in the USA. But Italy is surprising with the in vitro fertilization law, which was opposed for many years, which was really restrictive in terms of in vitro fertilization. But Italy is the first country in the world which unwillingly but openly opens up to CRISPR-Cas and to germline editing. In this law, it is said that the embryo is the subject of specific rights as all the other citizens are. So all genetic activities and all experimentations are not allowed, with the exception of those measures which aim at preserving the health of the embryo. If an embryo bears a specific disease and you can say that you can modify it to repair that gene, then you fall within the parameters of the law. If the legislator, according to certain values, turned the embryo into a subject which has the right to health, when CRISPR-Cas is available, then you make this kind of mutation of action possible. So Italy will be the first country where this is going to be possible by law, whereas in the United States, this is not possible. So in Italy, this can be done and will be promoted once CRISPR-Cas arrives, because this is really the result of ethical and scientific short circuit. I would like to close on this last slide, which I took from a conference which was organized last year by one of the inventors of CRISPR-Cas, who said that It's like a cursor in a Word file. It's a biochemical system telling you what you want to change the DNA. This applies when you put the mouse on a white piece of paper and you want to change a letter, for example. Far from discussing what this is going to happen, because it will happen. For example, in 20 years' time, we are going to have a child whose DNA is modified by CRISPR. It will happen, but we have to consider when it is going to happen, because we have to consider the values. We have to go behind the mouse, and we have to resist to self-correct, to autocorrect. In many cases, when we are sitting in front of the computer, of the iPhone, you want to send a text, but then you have the algorithms entering into play, and this is corrected, or self-corrected, auto-corrected. So we are corrected because the algorithm tells us that we wanted to think this, even if we wanted to say something else. But now we have a WhatsApp here. When we can change WhatsApp, we want to change it, and we want to be free to change it. That is why we want to have, and we must have, institutions which should not continue to discuss whether this is going to happen, because it will happen, but rather how it is going to happen. Because in a society which is unfair and unjust, we have not to translate this unjust approach into this modification of the DNA. This is the site. If you want to know more about what we are doing, this is the website. Thank you.